Our next speaker is Bart Rumsundar. He is the creator of an open source project called DeepChem, and he is also the CEO of Deep Forest Sciences. He's here today to tell us about ChemBERTa, which explores the use of BERT-style pre-training for machine learning problems inspired by drug discovery applications. Please welcome Bart Ramsundar.
Hello. Today I will be speaking to you folks about language-based pre-training for drug discovery. The work I'm going to be presenting on today is joint work with a number of collaborators at University of Toronto, Reverie Labs, and also done at my company, Deep Forest Sciences. The work is done as part of the DeepChem open source consortium, and you can find the paper online at Archive. ChemBERTa large-scale self-supervised pre-training for molecular property prediction. So before we start diving into specifics, I want to just lay the ground by discussing a bit about the biotech industry and pharma as a whole. So one trend that we've seen over the last several decades has been increasing costs. It's been more and more expensive to find a new medicine. This is due to a variety of factors. Some of it is due to increasing regulation for better safety for patients. Other possibly is due to low-hanging fruit getting plucked. So what's left are hard, challenging diseases, things like cancer, neurodegenerative diseases, where there's not a simple therapeutic strategy that's effective right now. And it's possible, there's a range of other factors that are also responsible for Ehrens law, but nevertheless, it's been a important reality that a lot of biotech and drug discovery companies have just had to deal with. And this kind of situation can be partially explained by the fact that therapeutic discovery is a long process. It takes a whole sequence of steps to go from a scientific idea to a medicine for a new disease. Typically, the starting point is something called target discovery. You try to come up with a mechanistic hypothesis for the root cause of a disease. And then you try to use that mechanistic hypothesis to identify a protein target, a biomolecule of interest that you'd like to drug with your potential drug. The next step in this process is typically what's called hit finding. Once you have this target, you just need to come up with some molecule that interacts with it in a reasonably selective fashion that doesn't go off target, so to speak, and interact with too many other biomolecules in the body. From there, you typically step-by-step optimize the molecule that you're working with, so it becomes less toxic, has better distributional capabilities, interacts even more tightly with the target you care about. And at the end, you finally are able to take this to something called animal testing, where you say, can I find an animal that has a version of this disease and cure the disease in that animal? If all this happens, then you're typically clear to start human trials. And then you also need to worry about things like process optimization. Can you manufacture enough of the molecule at scale so you can actually give it to human patients? One reality of this industry is that proprietary techniques are standard. If you ever talk to a lot of biotech or pharma folks, you'll hear the word, our proprietary technology, our secret sauce come up pretty often. And part of the reason for this is that biotech is kind of a long, relatively slow industry. Without patent protection, there's worry that copycats could come in and, basically make off with years of research. This focus on patent protection also extends to the software ecosystem for the biotech pharma world. Typically, software vendors retain tight control over their software systems. There's usually something like per-user site licenses. It's quite expensive, not easily accessible to students or hobbyists, which I think works well for funding complex scientific software development that takes a long time. But it does also mean that these techniques are not broadly accessible to, say, new graduate students or hobbyists or researchers who are outside, say, core top-tier universities. So these kind of background realities have also are coming to face with a new trend that's been rising over the last decade. It's what I'll call software-driven drug discovery. The core idea behind software-driven drug discovery is that discovery of new medicine is increasingly software-driven. Software-based guided design tools have started to become more popular in the industry. Molecular machine learning techniques, that is, machine learning applied to molecules, have become a robust and powerful class of algorithms that can really handle a lot of different data sets and conditions that are relevant for people working in biotech and pharmaceuticals. It's still early days for a lot of these techniques. Many of these new methods are only just now beginning to move from the research lab to the hands of practitioners. Another, of course, megatrend, which for this audience is probably very old hat, is that a lot of modern software is really driven by open source. So you have libraries like Postgres, React, TensorFlow, PyTorch, of course Linux itself, which are developed in the open by a large community of contributors who cross-pollinate ideas. So there's a bit of a tension here between the traditionally proprietary closed-source world of pharmaceutical drug discovery and the new kind of like open source software stack tech world. So this set of contradicting tensions was what pushed us towards starting the DeepChem project. So the core idea behind DeepChem is that open source can help medicine discovery by creating open source software tools for finding new medicine, by curating open source data sets, and by gathering a collection of educational material, that is books and tutorials. It becomes more easy to educate newcomers. So if you're interested, you can kind of find more information online about the DeepChem project at deepchem.io. We have a whole collection of tutorials, forums, documentation, places where you can get involved in our chat room. As a bit of prehistory about the project, I started DeepChem when I was doing my PhD several years ago. And from there, it's grown piece by piece as more and more contributors have found it useful for the types of problems that they're personally interested in. One of the core features of DeepChem is we maintain something called the MoleculeNet benchmark suite. This is a collection of different algorithms that, different data sets that make it easy to benchmark new algorithms for their efficacy and their predictive power. This ranges across a whole array of different topics, from quantum mechanics to physical chemistry, biophysics, physiology, and increasingly other areas like even material science. So with that background out of the way, let's now turn back to the focus of this conference, which is natural language processing. So I want to introduce you folks to something I call the Transfer Learning Grand Challenge for molecular machine learning. And the core idea here is that there's only a limited supply of labeled data about molecules. The reason for this really has to do with the fact that experiments are difficult to run. They're expensive, they take time. You have to be very careful about data curation, about quality control in order to get high quality data. This means that even the largest data sets with labels are in the hundreds of thousands of data points. Whereas if you go to unlabeled data, that is just repositories of molecular structures, you're able to get much larger data sets. There are collections of synthesizable molecules that range in the millions, even the billions. So one question here, of course, is there a way to leverage this large volume of unsupervised data to augment the power of our smaller labeled data sets? So, the answer here is that people have been trying to solve this problem for quite a while. There's been, a whole host of different techniques exploring different methods for doing transfer learning, where you bring in information from these unsupervised data sets to supervised data sets in question. There's also been a lot of techniques like, multitask learning, one-shot learning, and various semi-supervised learning architectures. And, the way I would summarize this entire field is that these techniques kind of work. It's a fuzzy word, but what I mean by that is that, there are papers, very good papers in some cases. They explain how to use these techniques, and in some cases even achieve quite solid results with them. However, none of these methods really is robust or works across many different data sets. And when we started this project, our question really was, can modern NLP techniques help us do effective transfer in drug discovery? Could this be an avenue towards a solution of the transfer learning grand challenge? So, of course, we've seen this absolute revolution in modern NLP with the rise of transformers. Things like BERT-style pretraining have really taken over a lot of different benchmarks and challenges in natural language processing. Again, for this community, this is all things that I don't really need to repeat, but you can, of course, see that more and more papers, more and more businesses are beginning to use these techniques in practice. So, in the rest of our talk, I'll introduce our new architecture. It's called ChemBERTa, continuing the series of BERT-based puns that tries to use these transformer-based techniques for challenges in molecular machine learning. So the first step here is that we need a way to adapt mass language modeling for chemistry. Typically the way that mass language modeling works is that you have, say, something like a series of tokens coming into an NLP model, and then you randomly mask something like 15% of the tokens that are coming in. And then you use the output to basically attempt to fill in the blanks on the masked words or tokens, depending on how you're doing this. So the question is, how can we adapt this to work on chemical data sets? So before we do that, let's just take a brief kind of pause and do some background looking at other methods on, pre-training for molecular representations. So this is a very cool paper that came out from Stanford a couple of years ago now, in fact. And the base idea is that you have graph-based pre-training. That is, similarly, you mask certain nodes that are in the graph. Then you attempt to do a fill-in where you predict, say, properties of the masked nodes. These can be their atom types, other embeddings. And the core concept here that we try to bring in with ChemBERTa is to merge these two ideas. We essentially mask out certain tokens in a language representation of molecules. This is called the Smiles representation. We see some examples over here on the right-hand side of Smiles strings. And then we use we use this mass language modeling technique to attempt to do fill-in. But implicitly, what we're doing is pretty similar to the Stanford SNAP approach in that the tokens we're masking are typically atoms or other important connections like bonds that are being represented in the Smiles string. So we are essentially doing a atom filling technique, as it's called, to learn about the structure of the molecule. To provide some more kind of context about where this project got started, one of our collaborators here on this paper, Cape Grant, he put together a first blog post called Training Transformers for Practical Drug Discovery with Tensor to Tensor as part of his work at Reverie Labs, a startup applying AI techniques to drug discovery. In parallel, some other members of our team through DeepChem had started evaluating different, ChemBERTa style architectures for application to drug discovery challenges. And we decided to kind of team up and bring these techniques together to see if we could, do something interesting. Before I dive into the details of our algorithms or techniques, I'll just say a couple results from our paper out on arXiv preprint from a few months ago. As of right now, these techniques, when you do pre-training with this mass language modeling technique and then testing on downstream MoleculeNet benchmarks, are functional, but not really at state of the art. So the DMPNNs are one of the stronger models recently published in the literature. And if you kind of check out the table down here, you see that at present, the ChemBERTa model, pre-trained on a collection of 10 million unsupervised compounds, is able to, it learns something, but it's really not fully comparable with the DMPNN approach. But, we did see some really intriguing results when we were doing this analysis, in that what we found was that as we did systematic tests on the the size of the unsupervised data set we used for mass language modeling, we saw downstream improvements in all of our downstream fine-tuning tasks. So this result kind of gave us an indication that our focus on supervised pre-training methods could actually be a powerful method. We only tested in this paper up to something like 10 million compounds for pre-training. But if you can scale that out to 100 million, to 1 billion or even more, it's potential that you could see, seems likely that you could see these curves continue to increase and increase, and demonstrating to us that we're on potentially a powerful strategy to improving performance in molecular machine learning tasks. So, there's a couple of things that we have to do on the tokenization end in order to be able to apply these ChemBERTa style techniques. When we first got started, we applied a byte pair encoding strategy. This is kind of a greedy strategy where you attempt to essentially take pairs of tokens and merge them into a single token in order to compress the vocabulary. This algorithm has the powerful advantage that you can reduce the length of the input sequences that are coming in. Some smile strings can get to be fairly long, and by applying this greedy strategy, you can get denser, more compact representations of the molecule that can be fed in to a network for mass language modeling. So in this case, we now have an example of how this byte pair encoding tokenization strategy is used on an actual molecule. So here we see that we have different series of tokens, or tokens in this case are in some cases single atoms, corresponding to carbon or sulfur. But in other cases, there are a series of tokens, like we have a CNC, carbon, nitrogen, carbon token. We have multiple, connectors, like, plus, left, right bracket, left parentheses, equal tokens. But this is roughly what's getting fed in as tokens into a ChemBERTa style model. We've also experimented with a new style of tokenizer. So this is something that uses the grammar of the Smiles language in order to better represent the molecule to the underlying machine learning system. So our tokenizer is based on previous work on the molecular transformer by Philippe Schwaller et al. And the core idea here that this work came up with was to use a regex pattern so that you could more meaningfully capture the chemistry at play. As you saw in our previous slide, some of the patterns captured didn't really satisfy coherent chemical constraints, whereas using the Smiles-Tokenizer allows tokens to be more chemically, semantically meaningful. This tokenizer is, open source as part of DeepChem, so you can check this out. If you look up the deepchem.feat.smiles-tokenizer, you'll see how the tokenization works, and we have like a brief code example here demonstrating how to apply the tokenizer on a new smile string. So, these are some kind of new unpublished results right now. So with this improved tokenization strategy, we find that we do see a nice boost in performance. So, broadly, we're more in the ballpark of the DMPNN method now by using this improved tokenization strategy. We need to continue testing our tokenization method at larger scales. If you notice, we've only pre-trained on 1 million compounds when doing this tokenization with the new Smiles-Tokenizer. So we're continuing to work on testing and scaling out these methods as part of our ongoing research. Another direction we've experimented with has been looking at new representations of compounds. So there's a new exciting paper called SELFIES where you use an alternate grammar to represent smile strings. And part of the challenge with the smile string is that you can have grammatically valid compounds that are semantically invalid in that they don't encode any known meaningful molecule. Whereas with selfies, grammatically valid strings correspond much more closely to chemically viable molecules. This means that for NLP-based methods, there's a much closer tie between syntax and semantics, which provides a powerful base for applying various generative methods to create molecules. So we wanted to see whether these techniques could enable improved performance of ChemBERTa. So this is ongoing. That's still ongoing work. It's still something we're exploring. Another idea that we are looking at is to use different pre-training strategies. So one strategy that has been common in the chemoinformatic literature is to predict various physicochemical descriptors of a molecule as part of the pre-training task. So here's a example of some of the types of properties that you might seek to predict. Some things like the number of radical electrons, the number of valence electrons, and so on and so forth for a molecule. This has been explored by a few papers. I think that we have a couple of citations here. And this goes back in the literature to a number of different works that have attempted to use this strategy. So we'd like to see if we can use this type of physicochemical descriptor to augment the pre-training in addition to the mass language modeling methods we've been using so far. We've also done some work on visualizations. We're trying to understand how our methods actually make the predictions that they do. So we've been able to use various highlighting techniques to be able to look at core atoms that contribute to our model predictions. So, using the BERT visualization, BERT-style library, we were able to look at some of the, core neurons in our network and see what they're learning. So as the video here on left shows, you're able to actually find some linkages in kind of representing what neurons in the model are learning. So an important aspect of our work is that, since it's done as part of a open source consortium under DeepChem, we've worked hard to make our work open and available to the community. The tokenizers have been open sourced as part of DeepChem, as I mentioned previously. Model weights have been open sourced as part of Hugging Face. So far, the ChemBERTa models have been called over 250,000 times using the Hugging Face API. So we're starting to see some pretty interesting broad usage of these algorithms and techniques out in the literature and community. We've also put together a code tutorial walkthrough as part of DeepChem. So part of what we try to do with DeepChem is we create a series of tutorials that explain various foundational concepts in chemoinformatics, bioinformatics, deep learning, and otherwise. So we put together a tutorial that tries to explain the core ideas behind ChemBERTa, and that lets you or anyone else really create their own ChemBERTa architectures to really learn about how these techniques work and apply it in practice. So, concluding the talk, there's a lot of reasons we think to be excited about these techniques for molecular machine learning. The kind of biggest one, of course, is over here on the right, where we see this semi-supervised learning effect, whereas we pre-train on larger and larger data sets, we see nice downstream improvements. So in our ongoing research, we're working on improving in the pre-training strategies. We're working on continuing to scale pre-training on larger and larger data sets. We're looking at doing more rigorous benchmarking. So we've only benchmarked on a subset of the MoleculeNet benchmark suite, something like 3 or 4 tasks. We'd ideally like to benchmark on all of MoleculeNet, which is 17 tasks in the first release, probably something like 30 to 40 tasks in the next release. We'd like to do more, systematic hyperparameter tuning. We'd also like to look at, techniques like graph transformers. There's some really intriguing linkages between transformers and graph-based networks. There was a paper called Grover and Europe's 2020 that investigated some graph transformer-based techniques for molecular machine learning that was particularly intriguing. So we're hard at work investigating these new directions. And with that concludes our talk. Thank you for listening. This work has been done by kind of a broad group of people under the nonprofit auspices of the DeepChem project. But our lead author, Seon, is actually still at time for speaking a high school student. We also have a number of engineers from Brewery Labs who've built out a lot of the core infrastructure, techniques, and ideas here. And of course, my company, Deep Forest Sciences, where we build AI techniques for use in different deep tech applications. And yeah, thank you all for tuning in. That's it.